With due permission of Dr. O.P. Bishnoi, Dr. Vijay, please start the session.
Yes, sir. The future of humanity depends not on what we consume today, but on how wisely we cultivate tomorrow. Good evening and very warm welcome to you all to day 12 of Agri SAARTHI KRANTI 2026, our prestigious 30 days National Agriculture Training Program. On behalf of entire Agri SAARTHI team, I extend a heartfelt welcome to all the participants, dignitaries, and esteemed resource person joining us today. Myself, Pratiksha, and along with me, Dr. Vijay Kumari ma'am will also be coordinating today's session. To begin with, I extend my respectful welcome to our course director, Dr. O.P. Chaudhary, sir. I also warmly welcome our organizing director, Mr. Ajay Kasana, sir, Chief Secretary of Agri SAARTHI, for his visionary leadership and unwavering support. Before we move ahead, let us briefly reflect on yesterday's enriching session by Dr. Sunil Dutt, sir, a veterinary surgeon, on dairy startups in India. The session highlighted the immense potential of dairy-based enterprises in creating employment opportunities, income generation, and rural empowerment, inspiring many aspiring agri entrepreneurs. Building on that strong foundation, today's session focuses on genetic improvement of bread wheat. In the face of climate change, evolving pest and disease pressure, and the growing demand for food and nutritional security, advances in wheat genetics plays a crucial role in developing high-yielding, climate-resilient, and sustainable wheat varieties for the future. It is now my privilege to introduce our esteemed resource person, Dr. O.P. Bishnoi, sir, Senior Wheat Breeder at CCS, Haryana Agriculture University, Hisar. Dr. Bishnoi, sir, has over 32 years of experience in wheat breeding and has been specializing in this field since 1994. He completed his BSc, MSc, and PhD from CCS HAU Hisar and was awarded the Best PhD Thesis Award for his outstanding work on terminal heat stress tolerance in wheat. Dr. Bishnoi sir has made remarkable contribution to Indian agriculture through the development of 16 wheat varieties suited to different sowing conditions across India. He also represented India at several international conferences and seminars, reflecting his global scientific exposure. Dr. Bishnoi sir's vast experience, scientific insight, and farmer-centric innovation will help us understand how research-driven breeding, genetic improvement, and sustainable practices can shape the future of bread wheat cultivation in India. With this positive spirit, I respectfully welcome and will invite Dr. O.P. Bishnoi sir to share his valuable insight with us. Over to you, sir.
हाँ जी डॉक्टर साहब,
आ रही है आवाज क्या?
जी डॉक्टर साहब आवाज आ रही है।
हेलो,
हाँ जी डॉक्टर साहब आवाज आ रही है, अभी आप स्टार्ट कर
आ
सकते
रही है?
हैं। हाँ जी डॉक्टर साहब
कर
बिल्कुल
सकते
आवाज आ रही है। हाँ जी।
तो फर्स्ट ऑफ ऑल, थैंक यू ऑल फॉर joining this lecture. Good evening to all the participants. I am here to present the development of wheat program in India since its inception in Indian condition from Green Revolution till date. So I am starting from the genetic improvement of bread wheat. Wheat crop is grown throughout the world. In India, we grow the spring type of wheat, and in the European countries, there is winter type of wheat. What is the difference between the spring wheat and winter wheat? Winter wheat is of long duration, approximately 180 days up to 250 days. So that require chilling requirement for its grain development. After sowing, up to 1 month, it is covered with the ice, and when it met the chilling requirement, then it will start growing, and the yield potential is high. But per day productivity of India is more as compared to the other countries, because our wheat matures in 145 to 150 days, approximately 145 days. Is I am audible to all?
Yes, sir.
Yes, okay. Then we move further. So the winter wheat that requires the chilling requirement, if we grow that wheat in Indian condition, then it either will not come in the flowering. If it comes in the flowering, then there will be no seed setting. So we make crosses of the winter wheat with the spring wheat varieties of Indian conditions to transfer their genes for hardiness, winter hardiness, for more biomass, for stay green characteristics. So we can use those varieties, but when these becomes photo insensitive, then it is used in Indian conditions. So I am moving here that wheat that is belongs to the tribe Tritici, subtribe and family Poaceae gramineae. On this includes 600 to 700 genera, and approximately 10,000 species are there of wheat. And in Indian conditions, three types of wheat, spring wheat is grown, in which first is Triticum aestivum, hexaploid wheat having AABBDD genome. Move to the next slide, Vijaya.
Yes, sir.
Next slide of cultivated wheat. Next. Next one. Yes, this one. So three type of wheat is grown in Indian conditions. First is Triticum aestivum, that is bread wheat or common wheat, that is grown around 95% of the Indian conditions, that is hexaploid wheat AABBDD genome, having genome. Second is Triticum durum wheat or macaroni wheat, tetraploid type of wheat AABB genome. It occupies only 4% of the area under Indian condition, in particularly in the central India, Maharashtra, in Madhya Pradesh, Karnataka, Andhra Pradesh, and this wheat is of industrial type. Products are made from this, that is macaroni, pasta, noodles. These products are made from this Triticum durum, and this durum wheat have the beta carotene content, that is source of vitamin A, that can be next. Yes. Third type of wheat is Triticum dicoccum or amber wheat. That is also tetraploid AABB and occupies 1% of area, that is southern hill area. And this type of Triticum dicoccum, that is in the southern part of the Indian condition. Next. According to wheat is a major staple food of the world population and occupies about 21% of the total cultivated area, accounting for 35% of total food grain production at global level. In Indian condition, it is grown around 31 to 32 million hectares, and in Haryana particularly it is in 23 to 24 or 25,000 hectares. Though the productivity of the country is around 35 to 36, 3 to 5, 3.5 to 3.6 ton per hectare, and in case of productivity, the Haryana is at the second position, that is 47 to 48 quintal per hectare, and at first position the Punjab state, that is around 50 or crossing the 50. Next one. In India, the total states are divided into 5 to 6 zones according to the agroclimatic conditions, in which the first zone is North Hill Zone. In North Hill Zone, it takes Jammu, Kashmir, excluding Jammu and Kathua districts, Himachal Pradesh, Uttarakhand, and Sikkim. This is North Hill Zone, and its area around is 0.8 million hectare, or centers which are working under this North Hill Zone, that is Palampur, Bajora, Dholakua, etc. The mega region of the wheat is Northwestern Plain Zone. Northwestern Plain Zone includes Punjab, Haryana, Rajasthan, Western UP, Himachal Pradesh, Jammu and Kashmir, and Himachal Pradesh, the plain area. It accounts for around 11.1 million hectares. The major growing area or wheat bowl is this region. Here is the centers, different centers which are working: PAU Ludhiana, HAU Hisar, Durgapura Rajasthan, Modipuram, and Pantnagar. These are the states of the UP, Rajasthan, Haryana, and Punjab which are working on wheat, and they produced a considerable quantity of wheat in the central pool. Third is Northeastern Plain Zone of India. In Northeastern Plain Zone, Eastern Uttar Pradesh, Bihar, Jharkhand, West Bengal, Odisha, Assam, and plains of northeastern areas. This accounts for 9.5 million hectare, and the centers which are working for wheat improvement in this zone are Kanpur, Faizabad, Varanasi, Sabor, Ranchi, Kalyani, Cooch Bihar, Shillongani, and some other centers. They are working on this. The productivity of this zone is low due to the heat stress conditions and leaf blight and other powdery mildew and other diseases. So the major focus of the scientists or working on the wheat group is that this zone should be improved for further yield increase, so that the national productivity can be increased. Fourth zone is Central Zone. Central Zone includes Madhya Pradesh, Gujarat, Chhattisgarh, Kota, Udaipur division of Rajasthan, and Jhansi division of Western UP. It accounts for 5.2 million hectares, and the centers which are working on this is Kota, Udaipur, Vijaypur, Sagar, Junagadh, Gwalior, Rewa, Puarkheda, and Bilaspur. And this region faces the problem of high temperature. High temperature, and in case of biotic stress, it faces the brown rust problem more. So we have to develop the climate resilient varieties which can thrive better under the central zone. Next zone is peninsular zone. that is the part of Maharashtra, Karnataka, plains of Tamil Nadu and Andhra Pradesh. It accounts for 1.6 million hectares, and the centers which are working on this are Pune, Nifad, Mahabaleshwar and Dharwad. And the last one is southern hill zone, Nilgiri and Palni hills of the Tamil Nadu, that is very less area, 0.1 million hectare, that is dicocum type of wheat is grown there. So these 5, 6 zones, according to the agroclimatic conditions and their needs, they are the scientists of these different centers, they are working on for different biotic and abiotic stresses. So next one, view of the wheat genome. How the genomic constitution of this is: three type of wheat, that is bread wheat Triticum aestivum. It is hexaploid wheat with sets of basic number of chromosome, that is X is equal to 7, 6 is equal to 2 and is equal to 42. More specifically, bread wheat is an exoploid with its three genome originating from the hybridization of the related species. Conventionally, these genomes are known as the A, B, and D genome. And AABBDD is the genetic constitution of the hexaploid wheat. Bread wheat has 21 pair of homologous chromosomes composed of 7 homologous groups, that is A1, B1, D1, A2, B2, D2, likewise A7, B7, and D7. Polyploidy in wheat provides the opportunity for greater variability among certain traits by manipulating the number of doses of a particular gene. Though it is polyploidy, but it behaves like a diploid. So this was the just genetic constitution. For more details, only the genetics and the plant breeding students require this type of information. And this is how polyploidy occurs. Means during the meiosis, second the chromosome cannot pair, they are homologous. Otherwise, in chromosome number 5b, there is P locus which provides the homologous pairing. Otherwise, if this gene, if the site of 5px gene is mutated, then there will be a great problem and the world faces the hunger problem. So the nature has given such type of homologous pairing by which the fertilization occur and the seed formation is there in the next one. Next, how this bread wheat is originated originally. That is, first the diploid type of originator Triticum urartu, that is the source of A genome, that was crossed with the wild type of Triticum speltoid or Agelop speltoids. By continuous mutation or natural selection, this AB hybrid, then by chromosome doubling, it becomes AABB, Triticum dicocoides, a wild type of durum wheat. Then it was domesticated and through selection, it becomes the Triticum dicoccum, a cultivated durum variety. This cultivated durum variety AABB, that was tetraploid, that was crossed with the wild type Triticum torsai or Agelopscurosa, the source of D genome. D genome is for the providing the genes for the biotic and abiotic stasis. Thus, is through crossing this Triticum dicoccais and Triticum torsai, ABD, by chromosome doubling, it becomes the AABBDD, hexaploid bees. And mutation and natural selection, there was the several types of the hexaploid bees, that is Triticum spelta, Triticum malsa, Triticum aestivum, Triticum compactum, and Triticum sphaerococum. They have different type of traits, but maximum growing in Indian condition is Triticum aestivum, that is free threshing. And next one, Next one. Here you can say the genetic characterization of these 6 types of hexaploid wheat. Triticum spelta, that is genotypic constitution is QQCCSS. So kernels do not thresh out, grow in USA, Southwest Germany and Spain. Means threshing is very hard. Triticum balsa, that was late maturity, winter type of wheat, that maturity is very late. And Triticum aestivum, QQCCSS, free threshing, tough rakish is tough and threshing is easy, so it is mostly cultivated in Indian condition. Triticum compactum, means compact type of spike. Ear head is of compact type grown in USA. Triticum spherococcum, free threshing, and grains are of spherical type. Spherical type means round type. 3 main mutations came in wheat, due to which this was mostly cultivated bread wheat. Recessive gene of Q mutated to dominant type Q, by which the changes which comes that free threshing grain and tough rakish. If rakish is brittle, then the shattering will be more. So this type of natural phenomena by which the mutation occurs and the free threshing grains and tough rakish was obtained, by which this was more successful, that is Triticum aestivum is grown in most of the areas. Recessive C2 dominant C allele on the chromosome 2D, compact ear producing allele, means besides lax ear, compact type of ear was produced by this mutation, and more number of spikelets were there in the ear and more number of grains were in compact type of ear. Dominant S2 recessive S allele on chromosome 3, producing spherical grains shape. Otherwise, it was elongated, but it is oblong type or spherical type of grains were produced by this mutation. Above 3 major genes responsible for chief evolutionary change that have occurred in the polyploid hexaploid wheat. Swaminathan and Rao 1960 were given these all details. Next one. Breeding objective in wheat. What type of breeding objectives by which all over the India the scientists are working, that they want to develop such wheat varieties which have high yield potential. Second, stabilizing yield levels by breeding for biotic and abiotic tolerance, means they are developing wheat varieties that are resistant to different type of diseases and different type of abiotic stresses like heat stress, drought stress, salinity stress. And second objective is to improving the product by quality, means by increasing the protein content, by increasing the iron content, zinc content, so biofortified wheat varieties. Next one, scientific wheat breeding in India. Foundation of scientific breeding work in India was laid down by Albert Howard and Grebilla Howard in 1905 at the Empirical Agricultural Research Institute, PUSA, which nowadays that is IRI, which was initially it was Empirical Agricultural Research Institute, known as IRI, located at New Delhi. Rao in 2001 described the period of wheat improvement work done in India between 1905 to 1962 into 3 distinct phases. Phase 1, phase 2, and phase 3. Next one. Next one, phase 1. In phase 1, there was the development of wheat varieties Pusa 4, IP4, NP4, Pusa 6, These were the varieties. Pusa 4 variety was so popular that it won the first prize successfully from 1916 to 1920 in international grain exhibition as one of the best quality wheat. Around 1939, some good wheat varieties were produced from Kanpur Center, K13, K53, etc., Nagpur Center also, and Lailpur Center also, type 8, type 9, type 11. This was the first phase. Second phase, this phase consisted of the production of wheat varieties of NP series, NP 100, NP 120, NP 165, and PB C581, PB C591, and making crosses between promising pure line variety developed during this phase. Third phase was, this phase include the varieties produced by the hybridizing Indian wheat varieties with the exotic wheat varieties. Exotic wheat varieties, that is Thatcher, Chinese White, Federation, Gabo, etc. During this period, main emphasis was for the development of disease-resistant varieties in 1950. Varieties developed during this phase was NP series, NP 700, NP 800, New Pusa, PBC 228, PBC 253, Hybrid 65. These were the varieties. So no real breakthrough in the yield could be brought during the period, at the country overall wheat production could be increased up to 13.4 million metric tons by the 1965. Next one. Era of semi-dwarf wheat varieties. Means before the development of semi-dwarf wheat varieties, what was the scenario in Indian condition? That glassy fuzz and Dharma, Northern America and Japan selections, from that Turkey red, from Russia, then Norin 10, that was having the dwarfing gene, a Japanese race, was crossed with the Brewer, and Gaines and Neve Gaines were produced in 1950. Next one, this Norin 10 was further crossed with the Brewer wheat variety, semi-dwarf varieties. F2 material was generated, and Dr. N.E. Borlaug produced these F2 materials, and from these F2 material, the F3 lines and spring type of Mexico material was crossed with this, and selection was done. Selection 14 and 17 from these in 1962, Pictic, Penzamo, Gabo, Sonora 60, Sonora, and second 8156. These best lines were selected from the segregating materials. Next, from this Lerma Rojo, selection from the segregating material, that Kalyan Sona. Kalyan Sona was the wheat which was first introduced in the Indian condition, and this variety was high yield potential. This was photo insensitive. Its adaptability was very wide. Input responsive, disease resistance. Input responsive means higher doses of nitrogen fertilizers were given to this, and there were no lodging, and by this high yield was harnessed under Indian condition first time through this Kalyan Sona. Next, this this was the golden era in the history of wheat in the country in 1963. After 1963 in India, dwarf wheat potentials were exploited by starting work in four directions. In four directions, first was introduction of dwarf wheat from Mexico. Then second, selection from the segregating material that was brought from the cement. Use of mutation, that Sarbati Sonera, which Sonera, which was of the red grains, through mutation it was changes to amber color, and it was given the Sarbati Sonera 64. And extensive hybridization, Indian into exotic crosses were made, and the wheat varieties were developed. For example, SD 2009, Arjun, and WL Ludhiana से 711 and WH147. A milestone variety was developed by CCSHU in 1974-75, WH147, which was um, responsive to the nitrogenous fertilizer and give the higher yield. Is first it was released in the northwestern plain zone, then WH147 was spread to the central zone and other zones of the country. Kalyan, Sona and Sonalika scaled up wheat production from 13.4 million metric tons in 1966-7 to over 29 million metric tons in 1976-77. So it was a very high jump, and this was called as the Green Revolution in Indian condition due to the introduction of the semi-dwarf wheat varieties, which were responsive to the nitrogenous fertilizer and also were the resistant to the diseases. Next one. Analysis of the pedigree of the four most successful crosses during 1966 to 90. That is, these wheat varieties, wheat varieties, which were having the blood of Viri, Kos type, Attila type, these blood of these background were very important for the Indian conditions, that the Viri that was very well adapted, cause that was very important for the disease resistant, Attila that was very important for the quality point of view. So number of landraces parents in the background of these varieties were developed. Next one. Vijay. Next one. What were the breeding methods which were used for the improvement of the wheat breeding program? First, 1945 to 1985, conventional breeding was used. From 1986 to 1995, modified pedigree method was used. And in modified 1995 to 1998, modified bulk pedigree method was used. From 1998, continuous use is the selected bulk breeding method is used. Means first we make the cross, then F1 is grown, that is non-segregating generation. From F1 we grow the F2 space planting, and in the F2 each and every scientist use his skill to select the plants of his own desire. Means for semi-dwarf wart, for early nest, for disease resistance, for better grain, etc., for tailoring capacity. All these traits are considered and selection is done maximum in the F2 generation. After that, the generation is advanced and the traits like grain size, grain color, that comes in the later generation, that is F6, F8. Then bulking is done. From that bulking, we go to the primary trials, then micro trial, then station trial, and then coordinated trial. Likewise, in the development of a wheat variety, from crossing to the release, it takes around 12 to 15 years. Next one, shuttle breeding method. Shuttle breeding method is used generally to advance the generation or to screen the genotypes for the disease resistance. For example, in Indian condition, in the plain area, we harvest the wheat in the month of April, and in the first week of May, we take the seeds because the wheat have no dormancy gene. So we select the lines which are better. We grow these lines to the hilly area. In case of wheat, for screening the genotypes for yellow rust in the Lahore Spiti, in Dalang Maidan. We grow the genotypes there during the summer and screen our material. If we have 2,000 lines and we get only the 150 or 200 lines which are resistant to the yellow rust, we take their seeds and we use for further multiplication of these seeds. So subtle breeding is very important to reduce the genetic load, and we want to screen the genotypes in early generation at Lahore Spiti, and important material is saved, and that is further multiplied for the further evaluation. Main objective of subtle breeding is to combine input efficiency and input responsive of wheat germplasm using alternate sites of contrasting environmental condition to bred for drought tolerance for semi-arid region of the world. If we want to develop for heat tolerant, drought tolerant genotype or high temperature tolerant genotype, then we have to move for the Jabalpur site of the Madhya Pradesh, because that site has the high temperature, and if the genotype perform better there, then it will perform better all over the other places of the high temperature. Next one, main steps which are involved in this breeding program. Space planted F2 generation is raised under optimum condition of the fertility and moisture. Single plants are selected as in case of pedigree method. Selected plants are bulk harvested and grown in F3 and F4 generation under stress environment. In stress environment, so that the proper selection can be done. From F4, F5 and F6 generations are again raised under high moisture and high fertility condition to harness their potential, to know what type of potential is there in these genotypes. Selection is done and lines which perform best under both environmental conditions are selected and assessed in international trial for yieldability and drought tolerance. Subtle breeding program was initiated at DWR Karnal in 1995-96 to support the wheat research activities of the northwestern plain zone of the country, because the northwestern plain zone of the country is very important to feed the central pool. So the high yield potential harnessing is very important in this zone. Next one. Major germplasm utilization at Shimit wheat breeding program. These were some lines which were used as a donor source. Genetic resources, McFadden Hope, SR2 gene stem rust resistance gene was used, that was durable stem rust resistance. Stem rust is particularly known as the black rust, that is in the uh, central zone, and the peninsular zone, that is major disease. So the genes from this was used. Japanese, Norrington and Brewer from USA, these are the sources of the dwarfing genes. There are 19 type of dwarfing genes, but RST reduced height 1 and reduced height 2 dwarfing genes were maximum exploited for high yield, high input responsive, reduced lodging. So these two genes are mainly responsible for the grain revolution. However, RST3 gene is also that is having the pre-harvest sprouting tolerance in wheat, that can be used in the area where there is the frequent rainfall during the harvesting season. So nowadays this is also used for to developing the wheat varieties which are responsive to, which are tolerant to the pre-harvest sprouting tolerance. From Australia, Gabo, that is was used PPD photo period 1 and photo period 2 genes for photoperiodism. Expanding the germplasm used globally. Photo period insensitive wheat varieties are developed that can be grown in the wider area as compared to its origin. Similarly, other 1B, 1R, natural translocation was used, durable rust resistance, leaf rust, brown rust 34 was maximum utilized. Similarly, all toxicity tolerance genes were used. Four-year blight diseases, that is northwestern plain zone, northeastern plain zone, that is more for leaf blight, etc. Kernel bunt resistance, large grains, Agilops cucurbica, and synthetic wheat was used for that purpose. So these type of sources, donor sources were used to develop the wheat variety for different sowing conditions, for different photo-insensitive wheat varieties, for pre-harvest sprouting tolerance, for disease resistance, for kernel rot resistance. Next one, genetic control of some agronomic characters in wheat. Biomass, that is polygenic characters, maximum photosynthetic rate, quantum efficiency, respiration rate, photorespiration rate, etc. Yield potential, that is major genes that are controlling plant height, that is yield components, that number of effective tillers per meter length, grain size means 1000 grain weight, more harvest index, plant height, though plant height, more plant height is negative, but because it is the main reason for the lodging, but without increasing the height, we cannot increase the biomass also. So we also have to consider a compromise level at which the maximum biomass should be used. Adaptation, vernalization response, photoperiod response, earliness per se. Vernalization means the temperature requirement. Mean if the winter wheat is grown, that is required, the vernalization genes are there in this, and photoperiod responsive genes. So our wheat is spring type of wheat, and the winter wheat, that is vernalization genes which are responsible for their chilling requirement. Yield limiting factors. Which are the yield limiting factors? That is disease, insect, pest, salt. They are the yield limiting factors. So we have to develop such wheat varieties which have the disease resistance, insect pest resistance, and salt tolerant wheat varieties. Adult plant resistant drought tolerance also consider. Quality point of view in wheat, protein quality, quantity, grain hardness. If the grain is more hard, then it is used for the industrial purpose, say for the durum wheat, that is used for the macaroni, pasta, noodles, etc. And protein quality in case of bread wheat. Next one, what are the strategies to improve yield potential? Improving the genetic yield potential. Improving the genetic yield potential. How can we increase the genetic yield potential? That is through broadening the genetic base. How can we broaden the genetic base? Uh, through the crossing or through exploitation of heterosis. Changing the plant architecture means erect type of leaves, drooping type of leaves, more number of tillers, long spike, short spike. Such type of architecture we can change by crossing the genotype. By technological intervention, we can also use the genetic engineering to improve the yield. Say, for example, if we want to improve for the salt tolerance, then we can transfer some genes from the salt tolerant plant to the well adapted variety. So by this way, the genetic barrier of crossing, that can be overcome by the genetic engineering. Reducing the losses due to the biotic and biotic stresses. Next improvement is how can we improve the genotype, that we have to reduce the loss to the through the biotic and abiotic factor. What are the biotic factors? In biotic factors, insect, pest, and diseases. Abiotic factors, say for heat, drought, and salinity. These 3-4 type of the abiotic stresses which are responsible for the limiting the wheat yield. So we have to develop such which varieties which are very tolerant to heat. Bridging the gap between the potential realized yield means the yield potential of some genotypes is much more, but at the real ground, there is we are harnessing the less yield. Say, for example, yield potential of the Punjab or Haryana, the average yield around 50 quintal per hectare, but the national level it is 35, 36 quintal per hectare. So this is the gap. We have to fill this gap, or we have to uh, narrow down this gap for improvement to increase the yield. Next one, pre-breeding to generative nib variability. How variability can be created? Means a plant breeder can improve the genotype or can develop the wheat variety of desirable type if he has the more and more variability in his hand. Variability is the prerequisite for any improvement program. So, to create new genetic variability, unexploited material germplasm like synthetic hexaploid developed at Simit by Mujib Kazi, long spike biter type means long earhead type developed by different scientists, Chinese subcompatoid ear germplasm, Chinese type that is of stay green type of genotype, more tillering capacity, have the genes for tolerating the low temperature as well as high temperature. So such type of genes we have to exploit for further improvement program. The main objective of pre-breeding are development of genetic stocks for yield component lines, supporting hybrid program by diversifying parents for heterosis breeding. So there is further chances because there is stagnation in the yield. For to break the stagnation in yield, we have to create the genetic variability. Next one, next one, features of different types of unexploited germplasm in wheat. You have heard that this is Triticum aestivum, this is Triticum durum, this is Triticum dicoccum, but there is synthetic hexaploid wheat, which is type of the synthetic hexaploid wheat that is having high grain weight, delayed senescence means stay-green type trait, high molecular weight gluten, resistance to kernel burnt and yellow rust. But there is problem with this synthetic wheat, that is they are hard threshing, red grain, delayed heading and maturity. Means maturity is so long, heading is also long, and red type of grains and threshing is very hard. So we have to improve. We can use these synthetic type of hexaploid in the breeding program to introduce the genes for the biotic and abiotic stresses and for quality point of view. Second is biter type of material. Biter type of material means long type ear head type genotypes or long ear type of plants. Robust stem. Stem is so robust that there is no lodging. Long spike, more spikelet, more grains per spike, large leaf area and broad leaves. Broader leaves, robust type of plant, strong stem, but there is lacking in this that these are the rust susceptible and shriveled seed because their duration is maturity time is so long that they do not mature in Indian condition in 145 or 160 days. They mature in 170 175 days. Then the due to the warm environment in the month of April, the grains are shriveled. So we can transfer only the stem, strong stem character and uh, long spike, ear length, and selection can be done in the next generation to improve our genotypes. Chinese subcompatible germplasm means robust stem, more grains per spike, new sources of yellow rust, very soft grains prone to insect damages, because soft type of grains are there in them, and the leaves are so less green that insect that attracts the insect pest. So we have to improve these our genotypes by crossing from these type of source material to create the new genetic variability and to improve our the well adapted variety. Next one, alien alien gene transfer. Alien gene transfer means transfer of uh, from the other sources, that Triticum timophilae, Agropyron elongatum, sickle cereal, and that is means critical type. These all have the biotic and abiotic stresses type of genotype characteristics. Several alien transfer involving small chromosomal segment or single gene translocations have made significant contribution to a development of new wheat varieties. Means by crossing these in the well adapted varieties, we can improve our genotypes for higher yield and disease resistance. Next one. There were at the time of Green Revolution various sources, say for Viri source, Caucas source, Attila source, 1BL, 1RS segment translocation. They improved the grain yield of 10 to 15% yield increase by improving the leaf rust resistance and winter hardiness and adaptation to different genotypes. Similarly, other translocations, they were also improved our genotypes and increased the yield. Next one, in 1995-96, there were 2-3 wheat varieties were developed which were of very important. WH542 from Wheat Hisar Center and PBW343 from the PAU Ludhiana Center. Our two wheat scientists, that is Dr. Mohammad Yunus from CCSHAU Hisar and Dr. Nanda from the PAU Ludhiana, they visited the cement center and made their selection. And after selection and in testing the material in Indian condition, the two wheat varieties were released simultaneously, WS542 and the PBW343, having high yield potential and disease resistance. However, they improved the yield potential of our condition a lot. But according to the time need, new and new wheat varieties were developed which are more higher yielder than these varieties nowadays, because PBW343, the resistance power breakdown for yellow rust. So the new wheat varieties were wanted again. Crossing of spring wheat with the winter type in 1917 and 98 has increased the biomass of spring wheat with increased yield potential as observed. Numerous cultivar released in many countries, Viri and Attila based. An important trait developed from synthetic wheat in the better root characteristics, means the root probably associated with the enhanced drought tolerance of synthetic wheat derivatives. Means when the synthetic wheat was crossed with the Indian well adapted varieties, then its drought tolerant component was increased, and which was used for the development of wheat varieties which are for the restricted irrigation condition, for the low input condition, say for WH1142 was released in 2015 from CCSHAU Hisar by involving the synthetic type of wheat derivatives. Next one. These are the some of the genetic improvement by development of hybrid varieties. Now, several type of efforts were made to develop the hybrid wheat, but we were not fully successful in this development of the hybrid wheat. Why? So I am trying to give some idea regarding this. The development of hybrid wheat varieties and thereby exploiting maximum degree of heterosis it plays a key role in achieving the goal. The effort of develop hybrid wheat in India were initiated 4 decades ago. Cytoplasmic male sterility in wheat. Kihara 1951 was the first scientist to report cytoplasmic controlled male sterility in wheat by transferring genome of Triticum aestivum into the cytoplasm of Aegilops cordata. Similarly, other scientists in 1953 transferred genome of Triticum durum into the cytoplasm of Aestivum ovata and developed a plant Aegilotricum. After backcrossing of this triticum, male sterile plants were developed. But these two types of cytoplasm could not be used for producing F1 hybrid because no suitable gene for fertility restorer were available. Restorer gene was not available, due to which we were not succeeded in the development of hybrid. Several, Likewise, several other scientists also attempted, but they were not succeeded. Myco, in collaboration with the Monsanto company, is the only company which has commercialized hybrid wheat based on cytoplasmic male style approach and has released 3 varieties: Pertham 7050, Pertham 7070, and New Gold for the central and eastern zone of India. These 3 wheat varieties were developed, hybrid wheat varieties were developed, but they were not much more successful in case of in comparison to our commercial wheat varieties. Why? This will be discussed in next one. Next one. Next one. Problems hybrid wheat seed production. What are the problems in the production of the hybrid wheat? Lack of genetic diversity among parents for harnessing the heterosis. There is heterosis is very less. That is less than 20% heterosis. For any hybrid, successful of any hybrid program, heterosis should be more than 20%. The maintenance of pure male sterility was very difficult in case of wheat. There was no restorer gene to restore the fertility in case of wheat. Pollinating capacity means nth type means closed flowering nature of wheat also is a barrier in development of hybrid wheat. There is synchronization in the male and female line is not the same, due to which the production of hybrid wheat is very less. Also, many wheat lines which were converted into male sterile lines after using chemophobic cytoplasm have wrinkled seeds. Their seeds were not good, and the and most and the last is that the hybrid seed requirement of per hectare is more in case of wheat. And when we compare these or evaluate these hybrids with our traditional breeding method developed variety, then there is no much more benefit. So up to now, these are not used in at commercial scale. Next one. Genetic improvement of bred wheat by mutation breeding. There were several type of to create the genetic variability. We use the X-rays, we use the chemical agents for to create the genetic variability by and at the time of green revolution, Sonera 60A that was having the red grain that was mutated with the gamma rays into the Sarvati Sonera 60. In 1967, amber color, grain color improvement was done. So Similarly, Lama Rosa also 1971 was improved by the mutation through from red grain to the amber grain. So a number of mutations and a mutation breeding program is going on to create the genetic variability for further utilization. Next one, there are several spontaneous mutations in nature also. That is also improving the wheat. More adaptation of wheat is due to the spontaneous mutation in nature. That is, so a large number of investigators have shown that emphasize that importation of natural mutation occurs in wheat. A major mutation which occurred in 5B chromosome has played a key role in the evolution in polyploid wheat. In 5B chromosome on the pH locus, there is a mutation due which the homologous pairing is there. Otherwise, there is very difficulty in the grain development or seed setting in the wheat. The mutation was for the diplodization of polyploid wheat. Mutation Q locus in wheat considerably affected threshability, means free threshing. Dwarf mutants also improved the RST1, RST2, which were main exploited during the Green Revolution for the responsiveness of the fertilizer uh, development wheat varieties. Next one, what are the constants in wheat production? Which are the factors which are limiting the yield level in wheat? Biotic stresses, particularly rust. Three type of rust, that is yellow rust, brown rust, and black rust. Yellow rust is the major disease of the northwestern plain zone of India, that is Punjab, Haryana, Rajasthan, Western UP, Himachal Pradesh, Jammu and Kashmir. That region is majorly affected by the yellow rust. If the wheat varieties are not developed for the yellow rust, and this rust infects a large number of area, and its loss may be up to 80% if the susceptible variety is grown. Though a large number of varieties by the different centers are developed which are resistance, having the yellow rust resistance gene, due to which there is a yield stagnation, yield stability in this zone. Second is leaf blight. Leaf blight, that is the major disease of the northeastern plain zone. Bihar, Kuch, Bihar, UP, West Bengal, that area affected by this leaf blight, peninsular and central zone also. And the stem rust or black rust, that is major disease of the southern part of the country, and have the major disease of the durum wheat and the estivum wheat also. And the central India, that is of high temperature, that is having the disease of brown rust. Brown rust, yellow rust. Yellow rust disease is of the major disease of the next northwestern plain zone, because at the time of growing season there is low temperature around 15 degree and humidity is high 80 to 90%. That is very congealing condition for the yellow rust. So this affects this northwestern plain zone. And the central zone, that is high temperature and high humidity, is required for the development of brown rust. So that is affected the central zone. High temperature and high humidity also in the peninsular zone, that is also affect the black rust and the stem rust. So these are the diseases and abiotic factors, particularly drought in the central and peninsular and northeastern plain zone. Heat stress, that is at the time terminal heat stress, that is at the time of maturity, that is northeastern plain zone is most affected by this. Salinity stress, that is uh, Uttar Pradesh and Punjab, Haryana and Gujarat. These areas are having the high salinity conditions, so this stress also limits the wheat production. Waterlogging condition. Though much more work has not been done up to the development of the waterlogged condition wheat varieties, though drought tolerant wheat varieties have been developed, heat tolerant wheat varieties have been developed, saline tolerant wheat varieties have been developed, but under for waterlogging, less work has been done up to now. Next one. These are the, as have already I told you that this is stem rust or black rust, that is warmer region, or SR2 and SR31. These are genes that was carried by the 1BL1R translocation, which is responsible for their disease resistance. Brown rust or leaf rust, that is was caused by Puccinia tritiana. Entire country is affected, particularly the central zone. Leaf rust resistance, these are the genes which are resistant genes. These are introgressed in the during the development of wheat varieties. For brown rust, yellow or stripe rust, Puccinia stipiformis, that is northwestern plain zone major disease. And these are the YR, yellow rust resistant 1 to 19 genes. These are the genes responsible for the disease resistance in the development. It is. Next one. These are the characteristics of leaf rust. Leaf rust, brown rust, that is high temperature, high humidity, and these are in the on the scattered form on the browning spots on the leaf, and they choke the photosynthetic site of the leaf, and there is no food production, and there will be shriveled grains will be produced. Next one, stem rust, that is also black rust, and it is having the SR31 or SR2 genes, and UG99, that is also a virulent race of the stem rust. For which we have developed a wheat variety WH1270, that is having the resistant against this UG99 and SR genes. So this disease is for the warmer and southern zone. Next one, this is the stem rust. On the stem there is black type of powder. Next one is the yellow rust. Yellow rust, I have already told you that this is the disease of the northwestern plain zone and major disease, and we have developed so many wheat varieties which are up to now. These are the resistant, say WH1105 wheat variety, 11 WH1124, WH1142, WH1270. These wheat varieties are resistant to for the yellow rust, and we also use them in the breeding program for further improvement of the well-adapted variety, which became the susceptible due to the change in the race. Next one, these are the sources of genetic sources by which from which we transfer the disease resistance. From having the disease resistance genes, and we use them as a parent and cross in the, and these are with the IC number. These are deposited in the NBPGR having the this type of parents, and we can use them for the breeding program. Next one, Karnal bunt. As the name indicate, this was first identified in the Karnal. Karnal bunt is caused by Newissia indica, first reported from India by Mitra 1931. Resistant donor for Karnal bunt is, there are 2-3 sources which are very resistant for the Karnal bunt. We used in the breeding program SD29, SD30 from the IRI PUSA, and Karnal bunt resistance lines are also developed from Louisiana. These can be used, and Karnal bunt has shown consistent decline due to the cultivation of wheat varieties PBW 343, PBW 5, PBW 17, PBW 550, and now a number of series of varieties are there which are resistant to kernel bunt. So a much more material is available with us for resistant to the kernel bunch. Next one, drought tolerance. For drought tolerance, we develop the wheat varieties which are very important for the those regions where there is less availability of water, the sandy soil is there, and underground water is salty. So we want to develop such wheat varieties which are very important for the sandy soils and less available of water in that area. For that purpose, different centers have developed so many wheat varieties which are tolerant to drought. Particularly, one old wheat variety C306 we have developed in 1965-69, high quality tall type, no dwarfing gene in this, but this variety have the um, very strong root system by which it sucks the water from the lower horizon of the soil, and it thrives well under the sandy soils and less irrigated soil. In in the same sequence, we have developed WH 1142 wheat variety, WH 402 wheat variety. So restricted irrigation, only two irrigation is required for that, for development of wheat variety. For developing wheat varieties, we have to consider two, 3 points. Number one, that wheat varieties should have a stronger root system, and stronger root system can be improved by using the synthetic wheat also. So such type of genotypes can be developed, which are well adapted to the under drought condition. Next one is heat stress. A major constant in limiting the heat yield is nowadays also is the heat stress. Heat stress is because day by day, due to global warming, the temperature is rising every year, and the cycle of the wheat crop or the duration of the wheat crop shrinks due to this heat stress. Sometimes in the area of the carrot, of potato, sugarcane, or due to the waterlogged condition, wheat is grown in the month of December. In the month of December, sown wheat that coincide with the high temperature at the time of maturity. At the time of maturity, there is less grain filling period is availed by the crop, then there will be shrink grains and there is yield reduction. So we have to develop such genotypes which have stay green characteristic, early vigor, short duration wheat variety, and more translocation in the developing grain at the time of heat stress. So they can maintain their 1,000 grain weight. So a number of wheat varieties by the different centers have been developed. We have also developed the um, strong, very heat tolerant wheat variety. One of our genetic stocks, WS 730, that is utilized by a number of wheat center for incorporation of the heat tolerant genes in the well adapted varieties. Next one, genetic base for high temperature tolerance is largely assessed by the monitoring the response of grain yield, grain filling duration in normal sowing condition. The grain filling period is around 6 weeks, means 40 to 45 days is required for the optimum grain filling period. But in case of December zone, this period reduced to 28 to 35 days, means 4 to 5 weeks. So there is less time for grain filling, and if the genotype have better translocation, then it maintains the 1,000 grain weight. Otherwise, there is reduction in the 1,000 grain weight, and ultimately the yield per unit area is reduced. Second, stay green characteristic is very important. More biomass is very important. Canopy temperature depression means some genotype they through transpiration they maintain their cooler canopy, and there is difference in the ambient temperature and the canopy temperature, such they maintain their temperature at the canopy level and better grain filling. So by this way they maintain their 1,000 grain weight and they give better yield. So a number of wheat varieties have been developed for the late sown condition for terminal heat stress tolerance by different centers, and University CCSHU SAR is also had developed a number of wheat varieties which are tolerant to terminal heat. Next one. After Green Revolution, first priority was to produce more and more food grain. After production of wheat varieties, our godowns were full from this wheat. Then we move to the improvement to the quality point of view. That which we produce, that should be of high quality so that it can be exported to the other countries and we can earn some money. So in case of bread wheat, which type of should be it? It should be hard, its gluten should be strong, protein should be more than 12%, and sedimentation would more than 60 ml. Chapati making, that it should not be so hard, it should be medium, gluten should be medium, protein should be less as compared to the bread wheat 10 to 12%, and the sedimentation value also 30 to 60. In case of biscuit making, we require the soft type of wheat. There should be weak gluten and protein content should be less than 10%, and sedimentation value also should be less than 30 ml. So nowadays we are moving more and more toward the quality point of view, and nowadays we develop the wheat varieties which are biofortified, that is having iron and zinc content more than 40 ppm, so that it can be um, of premium quality and it can be improve the our malnutrition problem of the below poverty line people. Next one. Classification of gluten protein contents, that is the same type of things. Means we have to improve our wheat varieties, particularly for the iron and zinc content, for high protein content, for low protein content. High protein content, more hard, that is for the industrial product, say for the durum wheat, which is very important for the pasta, macaroni, noodles, etc. And for biscuit making, we require the soft wheat, which have the less protein content and soft type of wheat. Next one. These are the chromosomal location of the storage protein genes of wheat endosperm. So it is not much required. Next one. These are the wheat promising wheat genotypes which were developed during means chapati making score. Total scale is 10, and the wheat variety which is having 8 or more than 8, that is of better quality. C306 have 8.6 of chapati making score, by which it is the best variety for the chapati point of view. In case of biscuit making varieties, that is 7.5 biscuit spread. Pasta making, that is having high protein content, we have our durum wheat variety WH896, and other centers varieties which are best for the pasta making and the macaroni noodles, etc. Next one. Breeding for biofortified wheat. Biofortified means the wheat varieties which have the high iron and zinc content. Scientific intake of minerals in the daily diet is essential for human health. Cereal grain contain low iron and zinc content. Almost all the semi-dwarf wheat varieties which were developed during the Green Revolution or up to now, they have the iron and zinc content up to 30 to 32 ppm. But some of the wheat varieties which have high iron and zinc content, that is 40 or more than 40, they are considered as the biofortified wheat varieties. Nowadays, the wheat varieties which of high yield potential are developed, they are also the bioforti type wheat varieties. Say for DBW 187 or WH1270, WH1080, WH1124, 4 5 these wheat varieties, these are having the iron and zinc content more than 40 ppm. So these can be used to mix the other wheat varieties to supply in the public distribution system to improve the malnutrition and anemia type problems in the weaker section of the our country. Next one, that is also iron. Next one, as I have shown that these WH1080, WH121, WH1105, these are wheat varieties which are having high iron and zinc content as compared to the traditional other wheat varieties. So these are considered in case of biofortified wheat varieties. Next one, raising yield potential trait based. Now we have to increase there is stagnation in yield. To break this barrier, we have to do something new. That we have to increase our biomass. Our wheat is C3 type of plant. So introduction of C4 like trait carbon concentrating mechanism, improving light interception through the semi-red type of leaves, optimizing spike and canopy photosynthesis, lodging resistance, sheep straw, optimizing harvest index, increase partnership, spike growth, and maximizing gram number per year head, more improving performance and regulation of Robisco. So by improving such type of activity, we can further increase our per unit area yield. Next one. Next one is we have to create genetic variability. That can be through the conventional breeding, that can be through biotechnological tools, that can be through mutation breeding. So we can, in case of biotech technology or genetic engineering, we have skill to transfer gene from the different crop, from different species in the well adapted variety to improve the terminal heat stress, to improve the salinity stress, to improve the biofortification, such type of traits, or we can transfer gene for the drought tolerance. So these fields are up to now not much exploited. So we can exploit these fields for further improvement and to develop high yielding and disease resistance and crop resilience varieties. Next one. Nowadays, the work is genomic selection. Recently demonstrated in CIMAT wheat breeding program, genomic based selection means in advance there is prediction on the basis of uh, plant type, so much type of greenness in the plant, so much number of tillers, so much height, such type of idiotype plant will give higher yield. So on the basis of genomic selections, there is nowadays we conduct a number of trials from the CIMAT which are based on the genomic selection based. On the basis of genomic, they are already proved that that will give the higher yield. But in our real ground level, we evaluate these wheat genotypes screened on the basis of genomic selection, and if they correlate with the high yield and disease resistance, then it is released as a new wheat varieties. And a number of lines every year are selected on the basis of genomic selections, and they also prove for high yield potential since last 2-3 years. Next one, these are the genetic stocks which are registered with the NBPGR having different type of sources for say for WH 4 to 3, that is high protein content, and also for the terminal it says. Similarly, other our wheat varieties, so many wheat varieties of STM and durum wheat varieties, they are of for different traits, which say for heat tolerance, say for multiple disease resistance, for high gluten content, sedimentation value, high iron and zinc content. So such type of work is done every year, and the list is enhanced every year by selecting better and better genotypes for further utilization. Next one. This source is old one. Nowadays, have a lot of wheat varieties have been developed. Uh, Next one, this is also somewhat old. It is not updated because the so many wheat varieties have been developed at the national level for different sowing conditions. Now, next one. Next one. Hmm. These are the wheat varieties, major wheat varieties which are developed from the CCSHAU SAARTHI since inception. That is 1965. That is C306 Desi wheat variety, which is the best wheat variety for chapati making score in the country. And the WH157 salinity tolerant, WH147 that is medium fertility condition, that is low input genotype, WH283 second quality wheat genotype developed by CCSHAR after C306, and WH291 that is for late sown condition, WH416 that is the improved group of WH147 having the lodging resistance, WH542 that is was released during the from the cement material and having the high yield potential and disease resistance, WH533 that is for the drought condition, rainfed condition, WHD 896 durum wheat variety, Sonak that is for late sown condition, WH912 for quality component, WH71 dwarf wheat variety which is high yielding, WH121 that is for the late sown condition. And these are the wheat varieties, landmark wheat varieties nowadays from CCSHAU is hard, WH1105 that is 100 centimeter height, lodging resistant, released in 2013, but it is still having the disease resistance for yellow and brown rust. WHD 948 that is durum wheat variety for the peninsular zone, that is released for the peninsular zone. WH1124 that is heat tolerant wheat variety. Heat tolerant. Next one. WH1124 that is heat tolerant wheat variety that is sown in the month of December and having good yield. WH1142 that is only for two restricted irrigation. And in 2021 a wheat variety was developed, WH1270 high yield potential through high yield potential trial. This variety was selected from a project, from German funded project, was that was for to develop wheat variety for early sowing condition, high temperature at the time of sowing, that is in the month of October, last week of October and first week of November. That is this variety WH1270 that can tolerate high temperature at the time of sowing up to 35 degrees centigrade. There is no effect on the germination and tillering capacity of this genotype. Similarly, WH420 1402 that is for restricted irrigation, low input genotype. Genotype means only two irrigation is required for this and high yield potential. WH420 two irrigation, it can give yield up to 50, average yield 50 quintal and potential yield up to 68 quintal. Similarly, WS1306, a wheat variety for better chapati making score after C306, that is released for the peninsular zone. Peninsular zone, Kerala, Tamil Nadu, and that region, and we are also trying to reach in the Haryana state. It has the chapati making score 8 and C306 is 8.6. So in our center, this is the second important mm, wheat variety which is best for the chapati making score, having the even the considerable yield level also. And WS1309, that is heat tolerant wheat variety released last year for the Haryana state, and it can be grown in the first week of the January, and it can give yield up to 55 quintal to 64 quintal per hectare. And these are the some of the photographs of the our landmark wheat variety WS1105. Next one, Vijaya. Next one. These are the wheat varieties. Some of the landmark wheat variety developed by CCS USAR WS1105 in 2013, and it is having the disease resistance up to now. WS1270 high yield potential, that is up to 19 quintal per hectare yield potential of this wheat variety. WS1124, that is heat tolerant wheat variety, and WS1402, two irrigation wheat variety that have threshed throughout the Rajasthan, and it is suitable for the sandy soil and low water requirement crop, and can give yield up to for average yield 50 quintal and potential yield up to 68 quintal. So in the last conclusion, the probable approaches for further increasing the yield potential of wheat varieties include: means we have reached up to 90 quintal per hectare, but for growing population we have further demand to improve the yield level. For that, what we have to do? Pre-breeding. Pre-breeding means we have to create the genetic variability through conventional breeding, through mutation, through other sources. Transferring of novel traits. Novel traits mean deep root system. Such type of traits we have to transfer from the synthetic wheat. Exploitation of heterosis means there is less heterosis, but if we exploit the heterosis, then we can develop the hybrid wheat. Breeding for drought and heat tolerance, as already discussed. Breeding for high tillering capacity. High tillering capacity means to increase the biomass, high grain number per spike. Vernalization or PPD genes means a photosensitive genotype we have to develop. An optimum level of PPD and vernalization in conjunction with the little larger maturity duration and high yield potential, that we have to transfer the stay green type of trait and other traits from these winter type of wheat into the spring type of wheat. Varieties like Viri represent Viri type background, cause type of background, Attila type of background, by advertising high protein line with the high yielding and disease resistance. Nowadays, we have to develop the wheat varieties which have the high yield potential as well as high quality traits also. In quality, protein content, iron content, zinc content, they have to, we have increased. Or there is a great demand up to 2050, we have to increase 60% yield total grain production. For that, we have to create the genetic variability, and we have to exploit the other sources of the creation of genetic variability, whether it is biotechnological tools, or whether it is mutation breeding, or whether we have to other sources to create the genetic variability. So next one. Next one. Next one. So thanks. Any question regarding this from audience side?
We express our deep sense of gratitude to our esteemed guest, Dr. O.P. Exceptionally insightful, inspiring, and knowledge enriching lecture on the genetic improvement of bread wheat. His vast experience and scholarly insight have greatly motivated and enlightened all the participants. Sir, thank you for this power packed session. We will now be moving on to the query and interaction session. Instruction to the participants: those who have question are kindly requested to use the raise hand feature. When invited, please unmute yourself, turn on your camera, briefly introduce yourself, and ask your query. Participants may also post their question in the chat box.
Good evening,
Okay.
sir.
Yes, please, Ritesh Kumar.
Sir, my doubt is you said there are no restorer genes which is used for producing CMS lines and producing fertility to hybrids, but there are some restorers line in wheat Indian varieties like NP at 39, NP at 83, and Kalyan Sona also derived from Kalyan Sona. Some variety like NP 150. Even some foreign variety like Prim Pevi, Palam Res, Marquis are some good source of fertility restorer lines. Um, there are so much restorer lines. फिर भी हाइब्रिड सीड प्रोडक्शन इतना डिफिकल्ट क्यों होता है?
Good question. आपने कहा yes, you have said that there are restorer genes. Though RF1, RF2, two type of restorer genes are there, but in Callistogenous nature of the wheat flower and pollen of wheat is not travel so easily. So there is a difficulty in the seed production. First, second, the seed requirement of wheat is much more per hectare. So if manually we produce some type of seed or through some rope we give the jerk and we transfer the pollen from the restorer parent to the recipient parent, then it is very difficult. So pollen travel is not so, it is not much success nowadays. And the second thing is that there is less yield increase over the conventional wheat varieties. So two, three hybrid were developed, but their shrink seed was there. Their yield gain was not much over the commercial wheat variety. So up to now, it is not success. In future, we are trying chemical hybridizing agents, which can there is some chances that by which we can improve our fertility level.
Sir, even nowadays, next generation chemical hybridizing agents like fenridazone, potassium, Hybrex, sorbitol compound, and some old chemical hybridizing agents like gibberellins, malic hydroxides are present, ethyl are present. Why doesn't that chemical hybridizing agents are used in producing male sterile lines?
There are some barriers in their utilization that we have already made some of efforts here. Our senior scientist, which have passed away now, Dr. Sudhir Kumar Shetty, CCHHU, sir, he has done a lot of work on that. At that time, I have joined there. We were we were we were making hybrids, but success was less. Nowadays, we have submitted a project for the hybrid seed production through the chemical analyzing agents. So we are further trying to produce hybrids by new way, chemical CHA by using that chemicals. So, though I have done a lot of work on the conventional breeding, not much more for biotechnological too, not much more for hybrid. Though I have seen their working, I have a part of them. I have a lot of experience of the conventional breeding, but future is of hybrid wheat or biotechnological tools. We have to improve because the yield barrier can be breakthrough. Only these two, three techniques are there by which we can improve.
Thank you, sir.
Thank you.
Thank you,
Any more?
Ritesh. Thank you, sir. The next question I call upon Ajay. Kindly unmute yourself and ask the question. Ajay.
Dr. Vijay, continue with someone else's question. He is not interested to ask.
Okay, sir. Participant, kindly ask the questions or ask your doubts. It's a golden chance to ask your questions.
Okay, I have gotten question from Ankit Malhotra.
Yes.
Ankit Malhotra is a postgraduate in soil science from PAU Ludhiana, and sir, his question is, can we expect in the coming years that the wheat crop can be grown in Kharif season also by breeding practices as a substitute for rice, and in Rabi season some other crop can be grown like pulses to diversify agriculture and to save our water resources?
Yes. In it is almost it is not possible that it is can be grown in the Kharif season, but we are extending our time that in the earlier time it was grown in the month of only November and December, but now we are able to grow the wheat in the month of October. In 15th of October, we have conducted trials in the month of October in a German funded project in which we can grow. There is variability in the genotypes, that is that they can withstand high temperature at the time of sowing in the month of October up to 35 degrees centigrade, and there is no effect on their germination. That is, their germination is 85 to 95%, and their tillering capacity was not affected. So variability is somewhere there in the genotypes, and if we try again and again, then its period can be enhanced. But in summer it is growing. Near future maybe some scientists like you uh, can improve furthermore, but chances is there that we can prepone 20 days, 25 days from the normal sowing condition.
Thank you,
Okay,
sir.
thank you, Naksa. Yes,
Sir, there
Pratisha,
is a
please continue.
question. Sir, there is a question from Ajay, sir. How to manage nutrients, weed, and management in wheat C306?
In C306, C306 is a low input requirement genotype, means it requires less fertilizer, less irrigation condition. So for growing such type of genotypes, every grower should kept in mind that he has to use less fertilizer. Say for example, 20 kg diammonium phosphate, 10 kg of zinc sulfate per acre at the time of sowing, and only 20 kg of nitrogen in the first irrigation, and only two irrigation have to be given, one at 30 to 35 days and second at 80 to 85 days, so that the plant will not lose and you can harvest the maximum yield potential. So any grower of C306 should kept in mind that he has to use either farmyard manure or vermicompost or less fertilizer and less irrigation for his crop production. By keeping these points in mind, he can harvest the maximum potential. Otherwise, there will be losing, and after losing, there will be less yield.
Thank you, sir. Next, I call upon Krishna Kumar Raghuamshi. Kindly unmute yourself and ask your question. Krishna Kumar Raghuamshi.
Hello, sir. Good evening.
Good evening.
Sir, my question is that is there use and role of integrated omics and digital tools in genetic improvement of wheat?
Digital tools, yes. Digital tools can be used in the wheat breeding program because a number of data has been generated through different crosses, through line into tester design, through generation mean analysis, through different type of mating designs, but that data is not fully exploited and fully interpreted by the different scientists due to the lack of digital interpretation. So nowadays these things have come. These things will further improve the our varietal development program. So I think that in future these digital data will help a lot in the development of wheat varieties.
Okay, sir. Thank you, sir.
Thank you.
Thank you, Krishna. Thank you, sir. Next question is from Jyoti. She is from Uttarakhand. Her question is, can India afford to prioritize quality-specific wheat when food security is still a concern?
Once repeat again.
Can India afford to prioritize quality-specific wheat when food security is still a concern?
Yes, food security is still concerned, but if the premium price is given, grower is assured that their product will be purchased at a higher price, then the farmer will move this side. And at today's there is situation that we have developed such type of wheat varieties which have very high yield potential up to 9 to 10 and ton per hectare, and even though they have the high quality components, say for iron and zinc content more than 40 ppm. So you can imagine that by broadening the genetic base we can increase yield as well as quality traits also.
Okay, thank you, sir. Next question is from Srikarni. She is UG student from Telangana. Her question is, in the area where there is a high salinity stress, we can use high fertilizer responsive varieties since they require high amount of urea application, which in turn increase the salinity of the soil.
Yes, you means that the genotypes which are tolerant to salinity, they have the salinity, whether it is drought tolerance, whether it is salinity tolerance. Once the genetic makeup of a plant is such type which have the adaptability to whether it is drought stress, whether it is salinity stress, then they can perform better under saline condition. And the high yield potential genotypes, they are developed for different soil condition, not for the salt affected soil and drought soil, because in the optimum environment the high input genotypes they perform better and they can give yield up to 9 to 10. And for salinity stress and drought stress, we have the different type of breeding program, and we develop the variety for that particular aspects, for salinity tolerance. Say for our Central Soil Salinity Research Institute, Karnal, had developed so many wheat varieties, KRL 210, KRL 213, KRL 283, 3, 4, 4, 5 wheat varieties. We have the better yield potential, as well as they are well adapted to the saline soil. So our aspect is different. Say for salinity, we develop the genotype for particularly for the salinity tolerant, not for high yield. High yield potential trial is for the optimum environmental condition.
Okay, thank you, sir. Next question is from Shivani. With increasing heat stress, drought, and new rust races, how can modern tools like genome selection and aligned gene transfer help in developing climate resilient bread wheat varieties?
Yes, genomic selection. Genomic selection is based on the genetic constitution in the genotypes, and which is say for every time we have 10 to 15 trials of South Asia bred wheat genomic yield prediction conduct trial, and these trials are formulated on the basis of their better yield potential under different sowing conditions, and they are evaluated under different environmental conditions. Say for Jabalpur, they are conducted for the drought tolerance. They are in the Uttar Pradesh, they are conducted for the terminal heat stress condition. In Indian condition also, they are in Haryana. These trials are also conducted in the month of December, so that the genotypes which were selected on the basis of genomic selection, whether they perform better or they are really heat tolerant, they are proved in the field also. So these things are correlated every year. So genomic prediction genotypes, there is chances that we can get the better heat tolerant varieties.
Thank you, sir. Next question is from Piyush Kumar. Describe the back cross breeding in wheat.
Yes.
Describe back cross breeding in wheat.
Back cross breeding. Back cross breeding. Back cross breeding is done particularly to remove some lacking point in a well-adapted variety. Say for our wheat variety, a well-adapted wheat variety is there, but there is that become susceptible, or resistance power is breakdown for the yellow rust. Then what we have to do? We want to improve that susceptible variety. For that susceptible variety, we take that variety as a recipient parent, and one wheat variety that have the disease resistance gene, yellow rust resistance gene, that will be used as a donor source. Once that is used as a male parent, and then in F1 and F2. In F2, we will select the genotypes or plants which are resistant type of genotypes. That will be selected, and that will be further we back after one back rows. If we want to back row second time, then okay. Otherwise, we every generation we select the resistant type of plant and go up to F6 or F7 generation. When the genotypes is in homozygous condition, then that variety developed by this method, that variety will be similar to the earlier susceptible variety, except in one trait, that will be resistant to the yellow rust or a particular disease. By this way, we develop the variety through back cross breeding, or back cross breeding is particularly used to remove some type of lacking, say for disease resistance or disease susceptible lacking is removed by this method. Any more?
ओके, आई हैव वन क्वेश्चन विद मी। डॉक्टर
Yes.
साहब, एक फार्मर है अपने साथ में, श्री कृष्णा सिंधे जी हैं, और ये पूछ रहे हैं कि कैन आई शो वीट ऑफ हीट टॉलरेंस इन फरवरी इन गिराठवाड़ा। ये गिराठवाड़ा कहाँ पड़ता है श्री कृष्ण सिंधे जी? हम तो यही नहीं जानते।
हाँ, कहाँ पे बता रहे हैं?
श्री कृष्ण जी, आप जो है हैंड रेज कीजिए, आपको अनम्यूट कर देते हैं, आप बेटर तरीके से पूछ पाएंगे।
हम्म।
चलो, टिल दैट टाइम अगर किसी और को
चलो
कोई
कोई
क्वेश्चन
बात
हो।
नहीं, बता देते हैं। इसका क्वेश्चन का यही जवाब है कि अब तक तो हम जो है ऐसी वैरायटी डेवलप करने में सफल हुए हैं जो फर्स्ट वीक ऑफ जनवरी तक सोइंग कर सकते हैं, और जिसका जो है 115 दिन के आसपास मैच्योरिटी पीरियड हो। लेकिन एक पेरेंट ऐसा सोर्स है जो हल्ला नाम की पुरानी वैरायटी है, इधर उत्तर प्रदेश यूपी कानपुर से डेवलप्ड है, वो 100 दिन के अंदर पक जाती है, 100 दिन के अंदर पक जाती है। तो उसके जीन जो है ट्रांसफर करके हम वी आर मूविंग फर्स्ट वीक टू सेकंड वीक एंड सो ऑन। एवरी टाइम वी आर ट्राइंग दैट वी कैन डेवलप some genotype which can only mature in 100 days. So and we have got success in a number of genotypes that we have developed a wheat variety WS1309 which can mature in 15 days and can give yield up to 50 quintal per hectare. So day by day, as the hurdles comes, we are trying to improve ourselves.
हेलो सर। हेलो।
हाँ जी।
हाँ सर, मैं मराठवाड़ा रीजन से हूँ, लातूर से सर। मैंने आपने
लातूर।
पूछा कि 3385 फेब्रुआरी में सोइंग किया था, तब भी उसका 10 क्विंटल निकल गया सर।
अच्छा अच्छा, 10 क्विंटल निकल गया था। थ्री।
हाँ 33, मैंने एक्सपेरिमेंट ट्रायल किया था राजबीर यादव से।
अच्छा 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 अच्छा।
तो इवन दो 40 डिग्री सेंटीग्रेड इट विल फील द ग्रेन्स, इट इज
फील्ड
माई
में
एक्सपीरियंस।
ये हो सकता है। तो हमारे भी यहाँ पे जो जीनोटाइप है अच्छे, आप वहां से लेके गए हो तो हमारा भी 1309 है, आगे लेके जाओ, आपके हो सकता है वहां पे ये अच्छा निकल जाए। 10 क्विंटल का कह रहे हो। हमने तो जनवरी में 10 फर्स्ट वीक ऑफ जनवरी में ग्रो करके 20 क्विंटल तक निकाल रहे हैं हम।
लेकिन ये जो मराठवाड़ा है ना सर, इसका टेम्परेचर आज 12 है तो कल 18 बन जाता है, ऐसा स्टेप बाय स्टेप बढ़ता नहीं सर, ऐसा जोन है हमारा।
अच्छा, अब अब्रप्ट चेंज आता है, एकदम ग्रेजुअल चेंज नहीं आता।
अब्रप्ट चेंजेस आता है सर। हाँ, तो इक्रिसेट
हाँ।
इसमें बहुत सक्सेसफुल हो गया है। रेड ग्राम का
अच्छा
45 डिग्री में भी अभी फीलिंग हो रहा है उसका।
अच्छा अच्छा, चलो अच्छी बात है। आपको हमारे को जानकारी बढ़ी, हमें भी अच्छा लगा। अच्छी बात है, और आगे भी हम भी इस तरह की।
मैं तो
हाँ।
मैंने ये नॉवेल एक्सपेरिमेंट किया, इनिशिएटिव किया है कि हमारा ऐसा अब्रप्ट चेंजेस होने के बाद भी मैंने वो 3385 हीट टॉलरेंट का बहुत अच्छा प्रोडक्शन दिया है फेब्रुआरी में। पहले मैंने
हाँ।
जो है, और अभी फेब्रुआरी में दिया था।
हाँ
थैंक
वो
यू सर।
थैंक यू थैंक यू।
थैंक
एक
यू सर।
लास्ट क्वेश्चन है डॉक्टर साहब, कृष्ण कुमार रघुवंशी जी का।
हाँ जी।
उनका क्वेश्चन है, हाउ डज जेनेटिक इम्प्रूवमेंट इन्फ्लुएंस न्यूट्रिएंट यूज एफिशिएंसी, नाइट्रोजन यूज एफिशिएंसी?
नाइट्रोजन की यस। नाउ अभी जो हम आगे हैं, नाउ वी आर मूविंग टुवर्ड दिस। वी हैव मेड ए कोलैबोरेशन विद द जापान. Our scientists have made that we have to increase the nutrient use efficiency. Means whichever fertilizer is applied in the soil, whether it is nitrogen, whether it is DAP, almost 33% is utilized. Other is either it is fixed or it is waste. Is audible?
Yes, sir.
Then now we are BNI one project is starting here. BNI biological nitrogen inhibition means we have to we are trying to develop such genotypes which restricts the utilization of fertilizer, means that they will use gradually, slow they will use the nitrogen, and biological nitrogen inhibition means the root system is such will be developed that will use less and less amount of nitrogen and phosphatic fertilizer to give the higher yield. Means by reducing the fertilizer level half, we can harvest the yield potential of up to 9 and 10 ton. Such type of research is we are now starting with the Japan.
Okay, thank you, sir. Extending to this question, I have one doubt, sir.
Yes.
Sir, actually, we all know that different crops uptake the nutrient in different forms. For example, rice uptake nitrogen in the form of ammonia, and also sugar cane also take uptake in the form of ammonia. Is it any gene is responsible for this kind of uptake, sir?
That is more depend upon the uh, condition of the plant growing. Say for in case of rice, that is the submerged condition. In submerged condition, ammonical form is required. In case of plain or in case of normal sowing, the plants they will take in the nitrate form.
Yes, sir.
In nitrate form, but there may be genetic variability in the genotypes also for their uptake, whether it is uptake ammonical form, whether it is uptake nitrate form. Less work has been done on this, and further future of this breeding program depends upon this nutrient use efficiency, that is BNI. Earlier I have said the biological nitrogen inhibition. So similar type of research is going on. We are moving towards that, and that will better will be told by the soil scientist, because he have the deep knowledge of these things. We are only concerned to the genetic variability in the genotype.
Thank you, sir.
Any more? So all have well interacted with, and thanks a lot to giving me the chance to interact with you and Ajay Kumar, and you both nicely conducted this session. Thanks a lot.
Thank you so much, Daksa. Dr. Vijaya, please conduct the and conclude the session for the vote of thanks.
Thank you, sir. On the behalf of Agri SAARTHI team, I would like to express my deep respect and gratitude to Dr. O.P. Bishnoi, sir, Senior Wheat Breeder, for graciously being with us today. Sir's distinguished presence and his vast expertise in the genetic improvement of the bread wheat added immense academic value to the session and inspired participants with practical and the research-oriented insights into a crop improvement and food security. My sincere thanks also to our organizing director, Mr. Ajay Kasana sir, Chief Secretary of Agri SAARTHI, for his continuous support, meticulous planning and guidance, which ensure the smooth and successful conduct of today's program. I take this opportunity to thank all the participants for their enthusiastic participation, active engagement, and thoughtful interactions, which truly made the session lively, meaningful, and successful. I also place on record my appreciation to the entire Agri SAARTHI team for their dedicated effort in organizing this 30-day National Agriculture Training Program and for consistently providing a valuable platform for learning, capacity building, and professional development. Before we conclude, I kindly request all the participants to ensure that the attendance form which has been shared in the chat box as well as in the group is duly filled. Please note that submission of the form is mandatory and the link will be remain active only 30 minutes. Once again, thank you all for your valuable time and participation. We look forward to your continued association and active participation in the forthcoming session of Agri KRANTI 2026. Wishing you all the pleasant and productive day ahead. Jai Hind.
Thank you.
Okay, thank you so much,
Thank
Dr.
you,
O.P.
sir.
Bishnoi,
हाँ,
to
ठीक है
bless our audience. And thank you
ठीक
so
है
much, all the participants, to listen to Dr. very patiently. So, thank you, everyone. Jai Hind.
Thank you.